Block 1, Unit 3, Drug Resistance in Cancer. Progress in the prevention, early diagnosis, and treatment of cancer has led to a recent decline in cancer rates in the United States. This advance reflects a greater awareness of environmental carcinogens such as tobacco, improved understanding of genetic contributions to cancer, more robust cancer screening programs, and for some cancers, improved treatments. Additionally, with an ever more comprehensive understanding of the genetic and molecular changes responsible for cancer growth and metastasis, a better appreciation of the interactions between tumors and their local tumor environment, scientists are beginning to decipher and target key pathways responsible for arresting programmed cell death, apoptosis, and stimulating cell division or metastasis. Targeting such pathways and understanding the mechanisms by which tumors evade current therapies should aid in the discovery and application of novel therapies. In fact, this approach has already produced drugs commonly used for cancer treatment, such as anti-angiogenesis, drugs that rob tumors of their blood supply, immunostimulatory drugs that boost the immune response against the tumor, and drugs targeted to protein changes resulting from specific genetic aberrations. Unfortunately, despite the tremendous progress made over the past 2 decades in the discovery of new therapies, drugs often stop being effective and ultimately, patients may succumb to the adverse effects of tumor growth or metastasis. In questioning why drug resistance occurs, our improved ability to characterize tumors at the DNA, mRNA, and protein levels leads to the conclusion that most cancers are heterogeneous, probably because of the high rate of mutation that occurs in cancer cells. There are approximately 200 different types of cancer based on tissue origin, and every time a new individual gets cancer, the precise set of genes that are involved may be unique. To borrow a paradigm that the director of National Institutes of Health, Dr. Elias Zerhouni, has applied to how modern medical research will revolutionize the practice of medicine, new approaches to cancer can be predictive, preventative, personalized, and participatory. Although not commonly used at present, molecular analysis of a specific cancer would better predict response to a specific therapy and enable tailoring of that therapy to a specific patient's cancer. Newer treatments will be preemptive by focusing on reducing the likelihood that cells undergo malignant change, and if and when this occurs, blocking growth of tumors so that they do not produce morbidity and mortality. This paradigm for the future of cancer therapy is now just coming to fruition. At present, definitive therapy in the form of surgery or radiation offers the best chance to cure patients with localized tumors. Blood malignancies and tumors believed to have spread beyond the primary site usually receive systemic treatments, chiefly chemotherapy and less frequently immunostimulatory drugs and anti-angiogenics. The new promises of targeted chemotherapy, striking at the cellular pathways that promote growth and metastasis or block apoptosis, is that therapy can be both personalized to account for tumor heterogeneity and much less toxic. So why, given our new arsenal of tools to treat cancer, are we still unable to cure patients with disseminated or metastatic cancers? Most people who die in the US with cancer have had chemotherapy that has failed, either because tumors simply do not respond to the therapy at levels that can be tolerated by the patient, or because cancers eventually become resistant to the treatment. Based on this fact, research has focused on listing and targeting general mechanisms of resistance to chemotherapy, one of the final frontiers in the conquest of cancer. Not all cancers are equal. They vary, they compete, and the fittest survive to pass on their genes to daughter cells, which continue to vary, compete, and survive. If this sounds familiar, it's because it is. Cancer cells evolve. Within the complex ecosystem that is our body, cancer cells mutate and face selective pressures as they change and adapt to their environment. The evolving cancer cell has to outcompete the normal cells surrounding it, even attack by immune cells, escape the apoptotic machinery which cause cells to self-destruct, corrupt and co opt otherwise loyal surrounding cells, and migrate to distant parts of the body. Sometimes, when the sequences of nucleotides called genes mutate, those mutations are not corrected. It doesn't happen often. Each nucleotide has only a 1 in 100 billionth to 1 in a billion chance of being miscopied. But each time a cell divides and passes a copy of its DNA to its daughter cell, the chance of error increases. An average gene comprises about 1,700 nucleotides, over 100 million billion cell divisions over the course of an average human lifetime. The average gene is thus home to somewhere between 100 million and 10 billion mutations spread over its copies in each cell. If just one of those mutations increases the evolutionary fitness of its cell, then the mutation will expand into many cells, increasing the probability that subsequent mutations will build on the first. We know that cancer occurs because we see the stepwise accumulation of mutations. If a cancer cell acquires a mutation that enables it to grow faster or survive longer and produce more offspring than the surrounding cells without the mutation, then the cancer cell has a selective advantage. Its descendants will be fitter. They will outgrow and dominate the local tissue environment. These principles of population genetics and evolutionary biology describe the process of tumor formation and are the key to understanding how cancer cells can become resistant to drug treatment. They evolve that resistance. Mutations enable cancer cells to embark on their frenzied growth within our bodies. But what are mutations and how do they happen? In genetics, a mutation is a change in an organism's DNA sequence. The nucleotide letters A, T, C, and G that make up our DNA can be deleted or substituted, and single or double-stranded breaks can occur in the DNA molecule. Complete sections of our DNA can also be deleted or swapped with other sections. These changes can occur spontaneously or from exposure to inducers such as harmful chemicals or radiation. Our metabolic activities cause mutations all the time. Oxygen, the vital molecule that helps us live, also creates dangerous DNA-damaging free radicals when metabolized by our cells. A sunny day at the beach can introduce thousands of mutations into our DNA. In fact, mutations are inevitable. Each time our cells divide, the imperfect DNA replication process introduces temporary errors into our DNA. It has been estimated that all of these processes can result in thousands of individual molecular lesions per cell per day. Our genome surveillance system and DNA repair mechanisms must do a fantastic job. Based on these mutation rates, cancer should occur from the moment we are conceived. What is our genetic surveillance system? If a cell is to survive, all of the different mutations must be repaired. Typically, this repair involves cutting out and resynthesizing the damaged portion of the DNA. Although there are many types of DNA repair, all are so important that the proteins involved can be found in everything from the humblest bacterium to our own cells. Initial chemotherapy of cancer usually begins with multiple cytotoxic drugs that kill cancer cells in different ways. The intent is to overcome resistance to any single agent, such as by mutation of the cellular target so that it no longer binds to the drug, and improve the likelihood of success. However, it was discovered in the 1970s in tissue culture systems that cancer cells are able, in a single step, to overcome the cytotoxic effects of multiple chemically unrelated compounds. This phenomenon, called multi-drug resistance, or MDR, may result from a variety of cellular adaptations, including reduced drug uptake, increased energy-dependent drug efflux, altered response of drugs because of changes to the program's cell death program, and altered cellular metabolism or sequestering of drugs within cellular compartments. Multi-drug resistance can be intrinsic. Cancers don't respond to any chemotherapy, or acquired, meaning that tumors respond, sometimes dramatically, but eventually grow back and are resistant to subsequent therapies. In most tissue culture studies of anti-cancer drug resistance, a predominant mechanism of both forms of multi-drug resistance is the overexpression of an energy-dependent transporter known as P-glycoprotein, the product of the MDR1 gene, or ABCB1 gene. This protein is a transmembrane transporter that resides in the plasma membrane of many cells, including cancer cells that are multi-drug resistant. The function of the protein is to move drugs out of the cell. They're therefore referred to as efflux pumps. P-glycoprotein recognizes a wide range of anti-cancer drugs, many of which are water-insoluble or lipophilic, natural products from plants, such as vinca alkaloids, paclitaxel, and fungal products, such as anthracyclines and actinomycin D, and keeps these drugs from accumulating at levels sufficient to kill the cell. P-glycoprotein is a member of the family of ATP-dependent transporters found in all living organisms that are called ABC, for ATP binding cassette transporters. In the human, there are 48 such transporters, and many are involved in the transport of lipophilic substances such as cholesterol and phospholipids into cellular organelles and out of the cell. A few of these transporters, notably ABCB1, ABCC1, and ABCG2, can pump a broad range of small molecules, many of which are used to treat cancer, and they've been found to confer multi-drug resistance to cancer cells. Several lines of evidence suggest that among these, ABCB1 is the most likely to be associated with clinical multi-drug resistance in cancer, including multiple myeloma, certain types of leukemia, breast cancer, and ovarian cancer. Since P-glycoprotein alone can mediate resistance to a whole array of drugs through drug efflux, it's an attractive target for the improvement of anti-cancer therapies. In theory, co-administration of transporter inhibitors, many exist, with P-glycoprotein substrate anti-cancer drugs could reverse MDR and improve treatment outcomes. Loss of the ABCB1 gene in mice, equivalent to the ABCB1 gene in humans, is compatible with life and does not result in an overt phenotype. These results have been interpreted to suggest that P-glycoprotein inhibitors would likely be safe for human use. Clinical trials aimed at specifically inhibiting the function of P-glycoprotein have given mixed results, but in at least some cases, this inhibition has resulted in improved tumor shrinkage and increased patient survival. Unfortunately, P-glycoprotein inhibitors, such as PSC833, induced pharmacokinetic interactions that limited drug clearance and metabolism of the concomitantly administered chemotherapy, thereby elevating plasma concentrations beyond acceptable toxicities. The solid tumors that have proved particularly untreatable, including colon, liver, kidney, and pancreatic cancers, tend to express ABCB1, but are probably protected by many other mechanisms of drug resistance, so simple intervention by inhibition of ABCB1 has not yet proved beneficial in treating those particular tumors. These results suggest the need for additional research in two areas. The first is to continue to identify novel mechanisms of multi-drug resistance, and the second is to devise new, more effective ways to combat multi-drug resistance due to ABC transporters such as P-glycoprotein. Research has taken bioinformatics approaches to the cataloging of cellular proteins that may be involved in multi-drug resistance in cancer, focusing first on those that determine levels of drug accumulation within cells, uptake inhibitors, and efflux pumps such as ABCB1. Researchers have taken advantage of the fact that the National Cancer Institute for the past 30 years has been determining the levels of resistance to almost 100,000 different cytotoxic compounds in 60 cancer cell lines, known as the NCI 60 cell lines, which include cell lines derived from many common human cancers. Knowing the levels of expression of the 48 human ABC transporters in these cells, and the level of expression of a variety of uptake transporters as well, has allowed us to correlate the likelihood that a given transporter is responsible for resistance, in the case of efflux pumps, or sensitivity, in the case of uptake transporters to a given drug. As a result, we have found that at least 28 ABC transporters are likely to transport specific chemotherapeutic drugs. Having such a list should expand the number of transporters whose levels should be determined in cancers in order to predict response to chemotherapy. Although it remains to be proved that quantifying levels of various uptake and efflux transporters will predict response to chemotherapy, clinical trials designed to test this hypothesis certainly seem warranted. One clear result of studies on drug resistance in ABC transporters is that transporters for virtually all of the anti-cancer drugs in current use, including new drugs that target specific growth-promoting pathways, have been identified. Since drug absorption, distribution, and excretion, pharmacokinetics, can be dramatically altered by ABC transporter function, the ability to predict transporter-mediated pharmacokinetic changes is of utmost importance. The normal function of ABCB1, for example, is to inhibit drug absorption from the gut, promote drug efflux from the liver and kidney, and protect so-called sanctuary sites such as the brain, testes, and fetus from potential toxins. Although normal expression of ABCB1 and other transporters can be considered protective, expression can also antagonize effective drug delivery. The wide variability in transporter expression and function relative to gene polymorphisms among individuals can also make it difficult to provide an optimal dose of chemotherapy. Notwithstanding these pharmacokinetic efforts, being able to predict drug transporter interactions should improve understanding of cellular drug resistance. In general, approaches can be taken, the 3 E's, to engage, exploit, and evade mechanisms of drug resistance. For ABCB transporters, this refers to the use of specific inhibitors of transport efflux systems that allow cytotoxic drugs to accumulate in cells, engage. The use of compounds that kill cells that express specific transporters, exploit. And the ambitious effort of the pharmaceutical industry to develop compounds that are not normally moved by transporters expressed in multidrug resistant cancer cells, evade.